As some states take the first steps toward trying to resume some type of normalcy, the risk of spreading the coronavirus infection remains high until there is a reliable vaccine. So how far are we from that? Joining us now is Dr. Ofer Levy. He's the director of the Precision Vaccines Program at Boston Children's Hospital, and Dr. Paul Goepfer, director of the Alabama Vaccine Research Clinic at the University of Alabama at Birmingham. Dr. Goepfer, I want to start with you. Paul Offit was quoted as saying the record for developing a vaccine is 4 years. 4 years, and that was with mumps. So when Dr. Fauci and others say a year to 18 months for COVID-19, really, how realistic is that?
Well, that's a good question, John. It is very optimistic because exactly our history is that we don't develop vaccines that quickly. This is an unprecedented time, and COVID-19 has sort of been on warp speed, and we're hoping that it'll happen more quickly. There are some things that would indicate it may be a little bit easier to develop a COVID-19 vaccine, but it's not for certain. And things such as the fact that the COVID-19 is not a very diverse virus, the fact that most people who develop infection actually get rid of the infection without major complications. Those things are actually very optimistic. That may lead to an optimistic indication that this could actually be developed quickly. But it is extremely optimistic to think that we're going to have a vaccine a year from now.
So, Dr. Levy, if you can, in basic terms that people as sort of dim-witted as me can understand, what are the stages in developing a vaccine?
Well, thanks for that, John. Typically, vaccines would be developed. The first step would be to define an antigen. An antigen is the part of the vaccine your body remembers after you inject it into somebody. And in fact, the term antigen comes from the fact that it is an antibody generating substance, an antigen. So you inject the antigen into somebody and their immune system recognizes it as foreign and makes, for example, antibodies against it. And those antibodies may protect against subsequent infection. So that's the basic concept of a vaccine. And all vaccines would typically have some sort of antigen in them. Now that antigen is typically, discovered in a laboratory, and then you find a way to make it, to synthesize it, and then you might test it, for example, in mice to find out if you inject the antigen into mice, do the mice make an antibody response against the antigen, and how high is that response, and how long does it last, and is it the kind of antibody that's really protective? These are all questions that scientists ask. And then eventually these kind of studies progress to larger animal models, and you might want to have a model where you can actually infect the animal with coronavirus, for example, and see if the vaccine would protect an animal against actual infection. And then if all of that looks good, you eventually work with FDA, the Food and Drug Administration, to get permission to get all the way to a phase 1 human trial.
And of course, you need to test it on humans to figure out, even if you can produce the antigen that will fight the virus, to figure out if there are any side effects. I want to ask you about antibody-dependent enhancement. This is something apparently where there's an extra threat for coronaviruses. Sometimes developing a vaccine for a coronavirus, which is what COVID-19 is more complicated because historically speaking, it's somehow produced a reaction in the body that leads to more infection. Can you explain that?
Yes, our immune system is smart. It recognizes a foreign threat and it will form antibodies against it to fight that threat. That said, what you hope to induce are the right kind of antibodies that not only bind very well, but dispose of the virus. If we get unlucky, if we don't have the right kind of antigen, there is a risk that we induce antibodies that may bind, but actually help the virus get into our cells. There are examples of this. There's an infection called dengue, and there was a vaccine against dengue that unfortunately induced antibodies that actually made the infection worse. So we're going to have to be very careful. There's a tension here. We want to move very quickly, and at the same time, we don't want to cause any harm.
So talk about the tension of trying to move quickly, because one of the things that people often push for is, look, the situation is so bad, we need to speed up the trial process. Yes, in a perfect world, we test it on mice and then humans and then more humans before we put it to market. But we have to skip some of those levels there. What's the risk of skipping certain levels or pushing it? For instance, the challenge level, where you might give it to people and then deliberately try to infect them. What's the risk of trying to skip that?
Well, there are inherent risks to any development of any medicine or vaccine. We've been there before. There have been, I'm one of the biggest proponents of vaccines, and the current vaccine schedule is excellent. Everybody should comply with it. I received my yearly flu vaccine and all my vaccines. That said, there are examples where vaccines, failed or on rare occasions even cause some harm. So we are really going to have to keep our eye on the ball. There are a lot of smart people in academia, government, and industry, unprecedented levels of collaboration now, trying to define the right path to strike that balance between speed and safety. Another thing to keep in mind is, even when we have an announcement, hopefully in the coming months, that appears safe and induces the right kind of antibodies, that will be a great moment for us, but we still are going to have to scale this solution to make hundreds of millions or billions of doses. That's not trivial. And I think part of the solution there will be to add an adjuvant, a molecule that may boost an immune response.
The scale thing is fascinating, right? Because normally we vaccinate newborn children, and that's in the several million in the United States a year. We're talking about vaccinating 300 million people here. It shows you why you need to be sure it's safe, but you also tells you about the problems in scaling.
Exactly. And I think we're going to be looking to, we're seeing a lot of innovation in the vaccine space. There are probably over 100 different groups developing vaccines, and we wish each and every one of them the very best. But we're going to be looking for innovation, but also we're going to be relying on the big pharma that has the experience for scaling to this degree. It will be unprecedented speed and scaling. Yeah.
Dr. Levy, we really appreciate you being with us and trying to manage our expectations here, what is possible, and we can all hope, what is realistic. And we also thank Dr. Ofer. We lost the technical connection there, so we appreciate your time.
Thank you so much, John.